Good afternoon. Thank you for giving us the opportunity to present ketamine 2022. I'm Marco Pappagallo, the Chief Medical Officer at REL-1017. We're excited to share data on the current development of REL-1017, esmethadone hydrochloride, in our presentation titled REL-1017, an NMDA receptor antagonist for the treatment of major depressive disorder. In this talk, we will discuss findings from our phase 2 clinical trial of REL-1017 for the treatment of major depressive disorder and provide our understanding of why and how REL-1017 is able to induce its antidepressant effect. Next slide. REL-1017 is a novel, low-potency, uncompetitive NMDA receptor channel blocker currently in phase 3 clinical trials for the treatment of major depressive disorder. Next slide. In our phase 2 trial completed in 2019, REL-1017 showed rapid and robust efficacy and a favorable safety, tolerability, and pharmacokinetic profile. The data from this trial was recently published in the American Journal of Psychiatry. Next slide. In this study, REL-1017 was investigated as an adjunctive treatment in patients who were experiencing inadequate response to a standard antidepressant therapy. This was a 7-day inpatient, randomized, double-blind, placebo-controlled study comparing REL-1017 25 milligrams and 50 milligrams to placebo. Patients were dosed in the first 7 days, followed for efficacy up to 14 days, and then for safety up to day 21. The safer interview was utilized after the initial screening to ensure patients entering the trial met the criteria for major depressive disorder to confirm quality results. The MADRS at baseline for all subjects was 34, a score categorizing patients as moderately to severely depressed. Baseline characteristics were well balanced across all 3 arms. Next slide. As you can see from the graph, both 25 and 50 milligram doses achieved statistically significant improvements compared to placebo, starting at day 4, then sustaining to day 7, and up to day 14, 7 days after discontinuation of REL-1017 at day 7. Differences in MADRS at day 7 were minus 8.7 for the 25 milligrams and minus 7.2 for the 50 milligram dose. And at day 14, minus 9.4 for the 25 milligram dose and minus 10.4 for the 50 milligram dose. For comparison, a typical NSSRI has about 3-point MADRS difference versus placebo. Another way to appreciate the efficacy findings is to look at the effect size. Here is as D. The effect size for the REL-1017 in this trial ranged from 0.7 to 1 during day 4 to day 14. Put this into context with other traditional antidepressant trials where the effect size are modest at best, around 0.34. Currently approved treatments can sometimes take as long as 8 weeks to show the onset of efficacy, where with REL-1017 we still will see a very rapid onset of efficacy. Next slide. Other important measures of efficacy are response rate and remission rate. The response rate is defined as at or more than 50% MADRS improvement from baseline. And remission rate is MADRS score of 10 or less at the endpoint. So both the 25 and 50 milligram doses demonstrated significant improvements versus placebo at day 14 for the response and remission rates. Next slide. So as far as the REL-1017 safety, there were no treatment emergent adverse events that led to patients dropping out or treatment discontinuation. Adverse events were infrequent. There were no serious adverse events or adverse event of special interest. Of relevance, no dissociative, psychotomimetic, or withdrawal effects, no euphoria, no respiratory depression were observed, and there were no instances of clinically meaningful QTC prolongation. Next slide. In addition to the safety findings from the phase 2 results, we recently completed 2 human abuse potential studies in recreational drug users, comparing 1017 to oxycodone, as shown on table 1 on the left-hand side, and to ketamine, as shown on table 2 on the right-hand side. A poster presenting these results are being shown at this conference. I'm here to provide a high-level overview of the key findings of these 2 studies. The primary study endpoint for both HAP studies was the drug liking, which at this moment, which is measured on a 0-100, a VAS scale, 0-100, bipolar, where 50 is neutral, 0 means means maximum dislike, and 100 is maximum liking. So the results of the REL-1017 versus oxycodone are shown here. We utilized three doses of REL-1017. The 25 milligrams, the 75 milligrams, and the 150 milligrams. The same 3 doses were utilized in the HAP study for ketamine. 25 milligrams is the therapeutic dose that we use in the phase 3 trial, and 75 milligrams 3 times the therapeutic dose. Also the loading dose on day 1. And 150 milligrams is the 6 times the therapeutic dose, and it's the maximum tolerated dose in phase 1 studies. We performed a completer analysis for the oxycodone. We had 47 completers, meaning that subjects who completed all the 5 treatments, and for the ketamine we had 51 completers, meaning that of subjects who completed all 6 treatments. For the ketamine, as you see, we also included dextromethorphan as an exploratory endpoint. For the doses, oxycodone was an oral dose of 40 milligrams, and ketamine was intravenous dose of 0.5 milligram kilo, given over 40 minutes. All the 3 REL-1017 doses were statistically significantly different and lower than oxycodone and ketamine, with a p-value of less than 0.001. And of note, the REL-1017 doses were statistically equivalent to placebo in both HAP studies for the oxycodone and the ketamine as well. So these results provide further support for our understanding of the safety profile of esmethadone or REL-1017. The drug did not show, in essence, meaningful abuse potential in both HAP studies. Next slide. So how and why does REL-1017 offer this unique property for the treatment of a major depressive disorder? Our preclinical work today assessed the effects of REL-1017 among other NMDA receptor antagonists, including dextromethorphan, memantine, ketamine, and MK801. Of interest, the work showed that REL-1017 has a preference for the N2D subtype of the receptor. And this might be an important novel variable in understanding its clinical action and its effects as an antidepressant. Next slide. So our hypothesis is that in major depressive disorder, the N2D-containing NMDA receptors may become tonically and pathologically hyperactive, and their dysregulated state would lead to an intracellular calcium overload and neurotoxicity in the affected neuronal population, with impairment of the physiological homeostasis of synaptic proteins and BDNF synthesis, ultimately impacting on neuronal and memory plasticity in the brain of the patient. Tonically and pathologically hyperactive N2D subtype channels may be a novel target for NMDA antagonists by downregulating excessive calcium signaling and positively impacting on neuronal and memory plasticity in the depressed brain. Essentially, we can restore the brain's ability to make new vital connections and neuroplasticity. Next slide. So what REL-1017 does is to repair the N2D containing channels hyperactivity by blocking the excessive tonic calcium influx, rekindle transcription and production of synaptic proteins, synthesis of BDNF, and ultimately promote the necessary neuronal plasticity that leads to recovery from major depressive disorder. It's of relevance. Our phase 1 studies have shown that REL-1017 can rapidly increase BDNF serum levels. Of interest also is the REL-1017 has characteristics of low receptor affinity and high traffic, which means, in our opinion, it correlates with rapid antidepressant effects with no dissociative symptoms. In contrast, ketamine has high potency and high traffic with strong antidepressant effects, but also with dissociative symptoms. Next slide. This slide here helps visualize the activity of the brain before and after the use of REL-1017. Of interest, the N2D containing channels among all subtypes have the highest probability of open configuration arresting membrane potential. So they are more likely to be tonically open and allow excessive calcium influx. The N2D containing channels do exhibit a higher glutamate affinity, a lower protection from magnesium blockade than other subunit receptors. So after treatment with REL-1017, the hyperactive N2D containing receptors return to operate physiologically, arresting membrane potential. Tonic calcium influx is normalized. Chronic excitotoxicity reverses, neuronal plasticity resumes, and depression recovers. The brain is then able to carry on the necessary connections, enabling major depressive disorder symptoms to go into remission. It's important to note that the development of REL-1017 for the treatment of major depressive disorder will contribute to the growing understanding and body of literature where now the N2D subtype may have an impact on several neurological conditions through its ability to normalize synaptic plasticity. Next slide. In conclusion, REL-1017 is dextroisomer methadone and a low-potency NMDA receptor antagonist. Preclinical data indicates a preferential action on the N2D subtypes, which may be the key pharmacological effect of the drug's ability to increase neuroplasticity and provide antidepressant effects. In our experience, REL-1017 does not induce dissociative effect, euphoria, or respiratory depression. It showed no meaningful abuse potential in the HAP studies conducted in recreational drug users of opioids and ketamine. So overall, we have a very favorable safety tolerability profile across all the phase 1-2 studies. As far as efficacy, we have seen a robust, rapid, sustained, statistically significant antidepressant effects on all tested scales. Of relevance, the REL-1017 is orally administered. It's once-a-day tablet. And finally, REL-1017 phase 3 clinical trials for the treatment of major depressive disorder are in progress. Next slide. So here are the list of references that we have used in creating our presentation and will make available to you for your perusal. Next slide. And lastly, thank you for listening to our presentation. Paolo and I will be available for Q&A for the next 10 minutes to answer all your questions you may have. We're looking forward to a good discussion. Thank you very much.
So, any questions that we can have?
Thank you.
This. Yes, Jason.
So I guess I'm interested in what has been done to characterize the NR2D selectivity and how different is that from the activity at some of the other sites?
Did you hear the question? What has been done to characterize the NR2D receptor and compared to other sites? Have we got them here?
Yeah, Paolo.
Paolo, can you
hear me?
we can't see Paolo yet. Great.
Okay. So what we did patch clamp experiments that we presented at the biological psychiatry meeting, and that's where we verified the preferential activity for the 2D subtypes. We were not surprised by that because there is literature that points out that this subtype is likely the preferential target of all uncompetitive NMDA antagonists because they all act physically by blocking the channel pore and reducing calcium influx. So that's, I believe, is a common mechanism of uncompetitive blockers. And what we see with ketamine is the Cmax-induced dissociation from a spillover of the block to a phasic activity block. So in other words, when ketamine reaches its highest concentrations at the doses used at the moment for depression, that's where we see dissociation. So and it's from our standpoint, we view that as a side effect and not part of the sustained antidepressant activity of ketamine, which is likely also due to this uncompetitive block, which is preferential for the 2D subunit, which is the subunit which is most sensitive to ambient glutamate and least sensitive to magnesium. So as you know, NMDAR receptors are coincidental receptors, and they require an open configuration, and they require being free of magnesium. They require these two things, and the 2D subtype is the subtype that's most likely to be in the open configuration with low ambient glutamate and free of magnesium. And we think that this is a requisite for activity at this receptor, because if the receptor is already blocked by magnesium and is in a closed configuration, you can flow through the receptor, and therefore there is nothing to regulate with an uncompetitive channel block. Sorry for the long answer, but open to questions still with Marco.
Is there a key experiment that you could do that would falsify your hypothesis that this was responsible, that this action at the NR2D was responsible for its antidepressant effect?
There is an interesting experiment that was, I'll provide the bibliography, an interesting experiment that was done with R-ketamine on knockout, 2D knockout mice. And what they saw there is that R-ketamine lost its sustained antidepressant effects in the 2D knockout mice compared to the wild type. So in other words, it was no longer effective when these mice had no 2D receptors. And I from our standpoint, it would be a support of this of our hypothesis, these findings.
I can add that there are also other experiments done with knockout the subunit 2D and 2D subunit, and these mice, they're totally, they act on the typical model of depression. They are totally inactive, depressed, in quotation.
In general, we believe that the 2D subtype permeability to calcium tonically is physiological. So this there is a percentage of these channels which are physiologically open during resting membrane potential, and this physiological calcium influx is likely necessary to maintain synaptic proteins, including BDNF, but also other synaptic proteins. Let's say the synaptic proteins that form the subunits of NMDA receptors. So
thank you very much.
Yeah.
Thank you very much indeed. That's very helpful. Thank you. Any other questions? Jonathan.
Hi, Jonathan Sporn. I just wondered if you could say two things. One is a little bit about what the frequency of the dosing and the design for your phase 3 study, because it seems like what happens after 2 weeks of in terms of maintaining patients, and then of course the issue, the concern always about abuse liability, given the scaffold it was great to see that it's differentiated from ketamine and from a strong opiate, but I wondered if there's any other data that would be useful in sort of kind of substantiating that lack of abuse potential, because I assume that'll be sort of a always a kind of concern just because of the scaffold.
Marco, maybe you.
Yeah, Well, so the phase 3 study is in progress, but so far we have no signal. And so the safety profile, from what we say, it's a blinded study, but we are monitoring, and we have obviously even the withdrawal aspect. So that we cannot really say much about that. The phase 2, this is what is the standard in industry and also in the literature are these HAP studies, and that for the abuse liability, the results are very clear. So that's one. We also have a preclinical data, traditional studies also very, very supportive with no signal. So what else can I say? I mean, this is now surprising. This, obviously with the dextro isomer potent other opioids are not really, they don't act clinically as opioids. The other thing is that there are also in the literature all studies done in the 40s, that's supporting this sort of our studies. So that's
okay. We're going to stop you there. Thank you very much indeed. That's really helpful. So thank you very much.